is this? Amoeba. Really close-up view of an amoeba, right? Because you're trying to get a view of this. What's this? The contractile vacuole. How does it move? What are these? What are these? Trypanosomes. Two diseases we've talked about in lecture. So what are the two? African sleeping sickness. How do they move? Flagella. You can pretty much see the flagella hanging off the end of these. That Giardia, obvious from this view, you can see it has flagella, right? It's not always so clear. And then, what do you call this stage when you're looking at this thing? A trophozoite, and the infectious stage is a cyst, right? That's from what? What's that? Spirogyra. What is it? Okay, well, so the spiral thing is a chloroplast. So, is it a plant? Is it a fungus? What is it? Not a fungus. Fungus don't have chloroplast. Algae, right? Yep, so it's an algae. Alga. And so, is this sexual or asexual? Not that. Sexual, what's the organism? Rhizopus, and what do you call this thing? The mycelium is like what you see, like when you pick up a piece of moldy bread and you see that mold on it, that's the mycelium. This is a name, zygospore. This is a zygospore. And that's, what places it in the zygomycetes group, because it makes this. And I think later on there's a picture of the asexual stage. This is hard to see, it's kind of small. But there's two colors here, so what is this? It's a gram stain. So which color is the gram positive?
Purple.
Purple, and the gram negatives are green. Okay, so what's the primary stain? What goes first? The crystal violet. What's the mordant? Iodine. What is a mordant? Here's
Fixes the
color. Yeah, it complexes it, so it makes it stick to what? Makes the crystal violet stick to the cell wall, right? And then you decolorize with Graham's decolorizer, which is an alcohol. And then what's the counter stain? Saffron. Okay, a little hard to see the colors. There's pink, there's blue. So what is this? Acid fast. So which ones are acid fast? Pink. Pink is acid fast. And what do you use to color it? What's the pink? Carbofection, right? And then, so what do you use for the blue? Methylene blue. Okay, so this one goes, primary stain is the carbofection. You then decolorize with something that's really harsh, acid alcohol, right? It's much harsher than Graham's decolorizer. And then you counter stain with methylene blue. Okay. So it says, name the genus of the bacteria that's bright pink. Microbacteria. So that's the TB and the leprosy, right? Alright, this one's dark, but can you see what color? That's supposed to be kind of a greenish color. And then there's supposed to be some pink in here, but some of this is really dark. This is the endospore stain. Okay, so what is the dye used to stain the endospores? Malachite green. And then the other dye, saffronin. And what is the saffronin actually staining? Not the cytoplasm. The cytoplasm of the cell. So like the vegetative cells have cytoplasm. They stain pink. The endospores will stain green for malachite. Name two genera of bacteria that produce the green structure. So that's kind of from lecture. The spore form, you know, Shubtail is not a spore form. Bacillus and Clostridium. Bacillus and Clostridium are the spore formers, and those see. Okay, so now the capsule stain. This is a capsule stain. Which organism were we using for that? The Liella as a capsule. So we used, which is the dye that we used in this one for the background? India ink, because we use nitrous and it just like washes away. So we have to use India ink. And then what was the dye that we used for the cytoplasm? Safranin. We use safranin. So safranin is the bacteria, and then the background is India ink, and then the white, the halo around it, is the capsule, which is not staining with either. Okay, so what is this? Granules for, and then who made this thing? Not aspergillus. This one is rhizopus. The aspergillus, you can see the edge is a lot more like bumpy because it's not enclosed in this membrane. Okay? Enclosed in the membrane is the rhizopus. Okay, this is a diagram. This is a diagram of the media that's testing oxygen requirements. So which media would it have? FTM. Okay, so if it's growing along the top, it's what? A strict arrow. And then your other choice was it could have grown throughout. A facultative anaerobe. Don't say facultative aerobe. Okay, facultative anaerobe. But in reality, there's other choices, right? We could have a microaerophile, which would have grown below the surface. And then, I don't really like this because the Clostridium doesn't grow just here. It grows all below the pink. So it would have been anything below the pink layer. The pink layer would have done this one down. So anything below the pink. So what was the pink? Okay, it was showing you where the oxygen was, and what was the indicator of that? Reasonant. And so it's pink when there is oxygen, and no color when there's not. So that's the same idea behind the gas pack, right? So the gas packs, we use the indicator. We use the same thing. We use reasonant. You can buy methylene blue strips, and they work the exact same way. There's a color when there's oxygen, and when there's no oxygen, there's no color. So you can just look after you've activated the jar and tanked it down after a little while, it should go colorless, right? Otherwise, you have a problem with your jar. Okay, we didn't use that name yet. We did this. So this is supposed to be blood agar plates, showing different types of hemolysis. So look at this one. What would you say is happening here? Gamma, right, there's nothing. There's no hemolysis in this first gamma. But look at this one. You can see the growth along the middle, and then there's this clear zone all the way around it. So what is this? Beta. And then what about that? Alpha. Okay, what about this one? EMB. This is EMB. So you can see the shiny green metallic, right? What is that telling you? Well, it is typical of E. coli. That's true. It is selective and differential media. That's true too. And so this is going to be a strong lactose fermentation. And this one is not doing that. But it's growing, so it's telling you something. It's telling you it's probably a gram-negative enteric organism because this is selective. What does it do? Yeah, it inhibits a lot of the others. So it inhibits gram-positives and some gram-negatives too. All right, this one is almost all yellow now, but it started out here, this pink red color. So what is this guy? Well, it's probably staph growing on what? Mannitol salt agar. So this is also what kind of media? Selective and differential. And so what's the selective part? It has salt, right? How much salt? 7.5. That's quite a bit of salt in it. Most organisms are inhibited by that, but not the staph. And the differential part is what? It has phenol red in it as the indicator of what? Acid from mannitol. So it's telling you it's fermenting mannitol. Okay, so yes, phenol red is that indicator. Because phenol red will turn yellow when there's acid present, right? And we're going to see phenol red again later in another test where it turns bright pink. So under alkaline conditions, it can turn a different color. So acid is yellow, then as it's more neutral, it's more red, and then we can get, when it's more alkaline, it turns more pink. Okay, this one is a little bit hard to see from the photograph, but it's been stained, and there's an organism growing here, and there's an organism growing here, and this has been stained with iodine. that you're using. Yeah, the starch plate, which the enzyme you're testing for. Amylase. And so this one doesn't have it, right? Because the iodine has stained all the starch right up to the growth. But this one actually does have a zone of clearing around it. It's kind of hard to see. The iodine floating at the top is looking kind of brown. But this has a zone of clearing around it. So this is telling you that the starch is gone. So that means the amylase broke it down. So this one is able to hydrolyze the starch. So it has amylase. Okay. Kind of small. If you see the small, sort of look on the plate. What are these little holes in the media? They're plaques. So what does a plaque represent? What happened? The bacteriophage is there, and what did it do? It lysed the E. coli that was there. So then you end up with a zone that's clear. Okay, so then this experiment was called a titer, right? We were trying to figure out the concentration, and just like you count colonies, this is time you count a plaque. We have plaque forming units, and we could figure out exactly how much phage was in the original stock culture that we started with. So know your term in the plaque form. A titer? So we have this long. Okay, what's that? Or trichomonas. It is. It's trichomonas, and there's actually 3 of them clumped together right here, along with some junk right there. So sometimes it's hard for students to see what you're looking at first. So this is a cell, like right here. Here's its nucleus, and here's its flagella coming off, and here's kind of flagella default. So we have a clump of cells here, but here's one individual. What does it cost? What? Yeah, it's an STD. So this is a protozoal STD. So trichomoniasis. So trichomonas is the word it was. That's an STD. Okay, so here's our friends, the micropipetters. You have to look at the scale and then look at what it's set to deliver down here. So let's start over here. So this one goes up to 1,000. So it says 200 to or 100 to 1,000 if you go on top. So if it's set for this, what is it going to deliver? 750 microliters. And so in mils, how much is that? Yeah, 0.75. Okay, this says 20 to 200. We use this one quite a bit. What's it set to deliver? 175. If you're under 75, what? Microliters. If you're going to set it, if you were going to report that in mils, how would you write it? 0.175. Okay, so this one says 2 to 20. I don't think we use this very often, except for maybe the first play with the pipetter experiment. So what is it set to deliver? 17.5. How would you write that in mils? 0.0175. Okay. And then what about this one? This one's small. This is a half, 0.5 microliter to 10 microliters. What's it going to deliver? 7.5. How do you write that in mils? 0.0075. So know your conversions. Make sure you know what a micropipetter will deliver, and then how you write it out in microliters and milliliters. All right, what's this? Radiolarians, right. How do you know they're not diatoms? Yeah, that's spiky, because they have these spikes sticking out. Okay, so do they photosynthesize? No, the diatoms do. These guys don't. These are like amoeba. Okay, they're amoeba with shells. So make sure you can recognize the difference between diatoms and radiolarians. What about that? What? Yeah, and what is that? What is that? It's a mushroom. So you're looking at, what are these things? These greenish things. Okay, is that the greenish thing or the red thing? The gills. The little red things are the basidiospores, and these are the gills, right? So if you flip a mushroom underneath, you see the gills. They're all filled along the edge with the basidiospores. Okay, so is this sexual or asexual? Sexual. Okay, so a little hard to see, but this is a gelatin deep, and it's actually kind of liquidy up on the top here throughout the slide that you can see it tilts a little liquidy, solid down at the bottom. All right, so what is the enzyme that you're looking for? It's a protease, right? So protease digests the gelatin up. So if it's liquid, it has it, and if it doesn't, If it's not liquid, then it doesn't happen. What tool do you use to inoculate it? Needle, inoculating needle. Okay, what test is that? Oxidase. And what is the oxidase testing for? What?
The final acceptor and electron transfer.
Yep, so you're looking at the final electron, the final member of the electron transport system in some organisms that transfers the electron to oxygen. And what's the name of it? Do you remember? Cytochrome oxidase.
What is the cytochrome oxidase?
Cytochrome oxidase. That's the oxidase. So is that positive?
Yeah.
Maybe.
It should be if it was something that happened fairly quickly after you did it, right? Not if you came back the next day and it was purple. That's okay. Okay, so what test is this? Indole. Okay, and so what do you add to test for indole? Covax. And if it's positive like this, what is that telling you? What's the biochemical activity it has? It breaks down
Covax?
which is an amino acid, right? So indole's made and you detect it with Covax. So positive, negative. Okay, so this, it tells you it's the methyl red test. Which one is positive? The red one. Okay, some people say the yellow one. So getting confused between the carbohydrate test or something else. So the methyl red test, when you add the methyl red, if it turns red, it's positive. What does that tell you about the organism if it's positive?
Mixed acid.
It's a mixed acid fermenter. So it takes glucose, it makes mixed acid, and the pH goes pretty low, goes below 5 to get that color. Okay, so this is methyl red. What's the media that you use to do this test? MR-VP. Methyl red, MR. Okay, methyl red, MR. And then the VP is which test? Okay, so here's the VP test. So it's the same media, right? You split it, VP test. Okay, so we add some reagents, and what color is positive? Yeah, this red one. Okay, and what were the reagents you add? A and B and ferrets, right? You add some ferret reagents, you get red. Okay? If it's positive. If it's positive, what does that tell you?
It uses the
Yep, it uses the butyl pathway for the glucose fermentation. And what it's actually detecting is acetylene. Okay, that's what's actually being detected. And if you're going through this right now and you're thinking, I don't know any of this, I don't remember any of it, it's not in your unknown report, go do that media DLA, right? It's in the STAR Center. And it has all of those tests from your unknown 1 report. It has the answer key, so if you go through your notes and you have holes in it, when you go to the answer key, fill them in, and then you're going to have a study guide that's going to help. Okay? So if I happen to make a comment on your unknown report, like this section is not a very good study guide, you probably want to go, do that DLA and double check the answer key carefully. Okay, what was this test? Citrate. So what does the citrate test tell you? Yep, so if it can use citrate as its only source of carbon, which result is positive? Blue. Okay, what happened? Why is it blue? The pH changed to up because of ammonia, right? It went basic because of ammonia. And why was there ammonia? You're testing citrate. Why is there ammonia? That's a urea. So the organism starts to grow, right? And it's using the citrate, but because it's using the citrate, it's also using the other things in the media, which is ammonium salts, and ammonia is released, and then you get the color change. So the color change is looking for, kind of like a side product of the growth. That tells you it's growing, so it can use sodium citrate. Okay? And the pH indicator was bromethymol. What about this one? Urease. Which one's positive? The pink. Okay, so now Alex, what happened? Yeah, urea becomes ammonia, the pH goes up, right? The ammonia is basic, and now what is the indicator? All red. So in this case, it actually started slightly acidic, and we get a rapid You get a hint here because you can see there's fluid down here, so you know something was added to this particular slant. And this one's a little bit green. Okay, so what test was this? Do you remember? The phenylalanine deaminase test, right? And so what did you add? What was the reagent that you added to it? Ferric chloride. So ferric chloride is the indicator. It turned green if it was positive. So then what? What did you know the organism could do? So the enzyme was phenylalanine deaminase. So what does that mean? Yeah, so it breaks apart phenylalanine, and a deaminase means, remember amino acids have a carboxyl group and an amino group, right? We looked at that back in the day when we were looking at amino acids. So a deaminator takes off the amino part. So that's what you're doing. You're splitting phenylalanine, you're taking the amino group off. So that gets detected here. All right, so what is this? Neutrophil. About how many neutrophils are there? Yeah, up to 70%. So this one is the most common, right? What's that? Eosinophil. Eosinophil. You know how much? 2 to 4? 2 to 6. 2 to 6? Is that right? Okay, what about a lymphocyte?
20%.
20 to 30. Wait, that's not a lymphocyte though. Now that I'm looking on the screen, this is a basophil. It's a basophil. So, how many?
Less than 1%.
Less than 1. Right. This is a basophil. There's a lymphocyte. Okay, what's this? Monocyte. Why do you know it's a monocyte? It looks like a big C, right? So this is really classic. Big fat C-shaped nucleus. That's another neutrophil. There's my lymphocyte. This is the 20 to 30. We cover all of them. We just did this one recently. Bioescalin. Not looking quite as dark as it. I don't know. The picture's not very good. Okay, so this one is black. This one is not. Which one's positive? Black one. What does that tell you? Okay, so it's characteristic of Enterococcus. It can split escalin in the presence of bile. Can most organisms split escalin? Yes. Yes, but not in the presence of bile. Only the few groups like Enterococcus. Okay, what was this? It's a small little plate.
Rodak.
It's a Rodak plate. What do you use it for? We're picking up things on
Surfaces?
flat surfaces, okay? So you're looking at the population of the aerobic organisms on flat surfaces. And you usually use it, you know, in places like after you disinfected something, did you do a good job? So you test it and you push this down and see if you actually got rid of everything that was there. Okay, what was that one? Coagulated. So this one has just some fluid. It's all, the tubes are tilted all the way out. It's going to be flushed out. This one is all body smug. So, what was in the tube? What's the media that clotted it?
Plasma.
Plasma. Which organism do we use this for? Staphylococcus. Okay, so there's a bunch of stuff here that says, look at this, look at this, you're going to do this. What is this? Water testing.
Oh,
Yeah, we use it for water testing. Most probable number. Okay, what's the little tube inside of the germ tube, and it traps what?
Gas.
Gas. And what was the media in here? We have the water. What are you testing for? Coliforms, right? So coliforms.